Okay folks, so we're going to talk about the enzyme linked immunosorbent assay, also known as ELISA. And ELISA generally takes place in a 96 well plate, and these plates contain 96 little holes or wells in the plate so that you can measure 96 different independent reactions as seen in the picture. But before we get on to the properties of ELISA, we have to cover the topic of antibodies, because the whole process of ELISA is based around the properties of antibodies. So as we've discussed before, antibodies have a specific affinity towards a given target, and I'm going to explain how that happens. So an antibody is made of two heavy chains and two light chains, seen with the blue being the light chain and the green being the blue being the heavy chain and the green being the light chain. But it's broken up into different parts. You have the Fab fragment, which is probably the most important fragment because it contains the antigen binding site. Okay, so the antigen binding site of different antibodies will be different because the antigen binding site is designed in a way that it specifically binds with its target. Whereas the FC region is a region that's generally well conserved. Okay, so all antibodies present in a human will have the same FC region structure. All antibodies in a mouse will have the same FC region, and so on. Now, in terms of antibody-based detection, what ELISA does is it measures the reaction of an antibody with a target that you're interested in, and it'll be a reaction that takes place in the wells of the plate. And the reason this is useful is that if we have a molecule of interest, for instance C-reactive protein, which is used clinically to determine if someone has an infection, is that we could take a blood sample and measure the amount of C-reactive protein present in that blood sample. And the way we would do that with ELISA is we would isolate an antibody that can specifically bind to C-reactive protein. And the whole concept of the ELISA is to try and isolate your target of interest from all the other molecules that are present in the blood sample. Okay. And the way we're able to do that is you have the antibody antigen or target reaction, and then because it's the antibody and antigen are rigidly bound to the plastic plate, we can then undertake a series of wash steps to remove the rest of the unwanted material, okay, all the other proteins in the blood. And what we then do is we quantify the amount of antibody and antigen bound through an enzymatic reaction. Okay, so what happens then is the degree at which the enzyme reacts or converts the substrate into a product will give us an indication of how much antigen or target's present. And generally what happens is the substrate and product will be different colors. So the change in color will give us an indication of how much antigen is present. And the color change is proportional to the concentration of your target. But how do we work out exactly how much material or how much target's present? We essentially create a serial dilution of pure target to create a standard curve. And when you create a standard curve, you can then compare the amount of color created from your blood sample to the standard curve and determine the concentration. Okay, so the intensity of the signal of the unknown sample is compared to the standard curve to get an actual value. And the concentration of the target in this case would be a blood sample, and in a clinical sample is determined. And once you get the concentration of your target, you then compare it to reference ranges and determine whether it is indicative of disease or of a certain clinical situation. Okay, folks, so in summary, the ELISA is a antigen antibody reaction that takes place in the plate that enables us to quantify a target of interest. But before we get into the specific examples of ELISA, we need to be clear on certain terminologies. Okay, so firstly we have a capture antibody. Capture antibody is usually polyclonal, and the concept of the capture antibody is to isolate and bind the antigen from the complex blood or other biological sample. Okay, so the capture antibody provides an avenue for us to bind the antigen and rigidly hold it down, immobilize it essentially. Now it must be noted that capture antibody is generally only used in a sandwich ELISA, and we'll come on to that in a little while. So whilst the capture antibody is there to isolate the target, a detection antibody, which is usually monoclonal, okay, so it's very specific antibody, is used to bind to the target for quantification purposes, okay. So in this example, this is sandwich ELISA, which we'll cover later, but here we have a capture antibody isolating the antigen, okay. But then we have the detection antibody that binds to the antigen specifically in order to subsequently quantify it. You can also have a secondary antibody, and the concept of the secondary antibody is to bind the detection antibody to facilitate the quantification. Okay, so here you can see the secondary antibody binds to the detection antibody. And the thing to note is the secondary antibody has an enzyme conjugated to it. And the whole concept here is that once we have this series of reactions, which will be different based on different ELISAs, but we'll cover that in the next few slides. But once you have this series of reactions, the way we're able to quantify it is through this reaction, where a substrate reacts with the enzyme to produce a product. And this reaction causes a color change, and the intensity of color change tells us how much antigen is present or how much target is present. Now, in terms of other terminology you may come across if you buy an ELISA kit, is it will refer to anti-species antibody. What this basically means is it tells us where these antibodies came from, i.e. was this antibody produced in a mouse, was it produced in a cow, and it'll tell us whether the antibody is anti-human. For instance, in this case, if you're measuring a target in a human, and the antibody that is directed against anti-human, for instance CRP, then it's referred to anti-human. Okay, so if I was to use an example, if this antibody was produced in a sheep directed against anti-human CRP, then it's referred to sheep anti-human CRP antibody. Okay, so in this case I've used insulin. Okay, and if the detection antibody was produced in a mouse, okay, if the detection antibody was produced in a mouse directed against anti-human CRP, then it's referred to as a mouse anti-human CRP antibody. And for instance, the difference here is that with a secondary antibody, it has to be targeted against the FC region of the detection antibody. Okay, so this antibody has a specific affinity for the conserved region of the antibody. So in terms of this nomenclature, or the way we would do this, is the first thing you consider is where was the antibody produced? Okay, where did we extract the antibody from? Okay, so in this case the antibody was produced or extracted from a sheep. The next thing is what species is the antibody targeting? Okay, In most cases, it'll be human, okay? So in the clinical setting, all antibodies will be directed against a human target in a clinical lab. And just to remember that the secondary antibody is always anti-human and it's always targeted at the Fc region, okay? So the secondary antibody will always target the Fc region of the antibody. So in terms of signal quantification, we talked about the fact that with enzymes you can get a color change in terms of quantification, but it can be measured through a whole range of things. It can also be fluorescence, chemiluminescence or electrochemiluminescence, and if it's a standard color change it'll be absorbance. You can also get other ways to quantify immunoassays such as fluorochromes, which we're not going to cover, and also you can use radiation in a radioimmunoassay. So now we're on to the actual ELISA methods, okay? So generally speaking, there are 4 types. You have the direct ELISA, the indirect ELISA, a sandwich ELISA, and a competitive ELISA. So the direct ELISA involves the presence, or generally speaking, involves the presence or absence of an antigen, okay? Indirect ELISAs are usually used to quantify antibodies within your blood, okay? So for instance, if you have produced an antibody against HIV, an indirect ELISA would be used to measure how much of that antibody you have produced. A sandwich ELISA is used to detect antigen, generally speaking. It can be direct or indirect, and we'll cover that. And lastly, you can have a competitive ELISA. So we're going to start off with a direct ELISA, okay? So in a direct ELISA, what happens is you add your sample containing the antigen target. The antigen target subsequently binds to the plastic plate, okay? Because you've isolated your target on the plastic plate, you simply then add on your detection antibody, and in this case the detection antibody has an enzyme attached to it, okay? And then you add your substrate and then your substrate, and you get an enzymatic reaction. So the direct ELISA is actually quite quick because the detection antibody has the enzyme attached to it. Whilst an indirect ELISA is the same concept, where you have the antigen, the company puts on the antigen and adheres to the plate, and then you add your blood sample, which contains an antibody that you're interested in, for instance an antibody against HIV, and when it binds to the antigen you then add a secondary antibody, okay? The secondary antibody binds to the Fc portion of the antibody from your blood, and it contains a secondary enzyme. And then at that stage you add the substrate and you get a signal color change. So I'm just going to take you through each one of individually again, okay? Another example would be with the use of a direct ELISA is where if you had a milk sample and you suspected that it might contain enterotoxin, which could be dangerous when consuming milk, you add the sample to the well, and of course there's a whole range of other components in the well. They all attach to the well, okay? So the protein components present in the milk sample attach to the well and become immobilized. The next thing that happens is you add your detection antibody, which is colored in purple, and the detection antibody binds to the target, and subsequently when you add the substrate, you then do a wash to remove it, and you add the substrate and you get a color change. So just to go over that again, you have a milk sample which you think is suspected of having contamination containing enterotoxin, which is dangerous to health. So you add the milk sample and a whole range of proteins and other components bind and adhere to the plate. Okay, they're immobilized. And what happens then is you add a detection antibody with an enzyme attached, which is directed against the enterotoxin, and it subsequently binds the enterotoxin. You then wash away the excess unbound detection antibody, and then you add your substrate. Substrate reacts to the enzyme and you get a colour change. Okay, now an indirect ELISA. So an indirect ELISA involves a primary antibody or a detection antibody, but it also involves a secondary antibody, usually that has a conjugated enzyme attached. And in terms of when an indirect ELISA would be used, a good example would be HIV infection. Okay, so people with HIV infection produce antibodies directed against HIV. Okay, and the presence of the antibody that's directed against the HIV that would be present in someone who's infected can be measured to give an indication of the presence of HIV. And indirect ELISAs are generally used to measure an antibody of interest that's present in the blood. So here we have an indirect ELISA. So generally speaking, an indirect ELISA, the company will provide the antigen attached to the plate. Okay, in this case, the company will attach the HIV virus antigen because we're interested in detecting the antibody. And generally speaking, what will happen is you then add your blood sample. Okay, and if it contains antibody direct against HIV, when you add it, the antibody will attach to the HIV, and the other antibody that's not directed against HIV will be washed off. So now we have HIV antibody immobilized in the plate because it's attached to the antigen. But at this stage we have no idea of how much antibody is present. So we then add a secondary antibody. Okay, and the secondary antibody then is targeted towards the Fc region of the target antibody. When it binds, you get the secondary antibody binding to the antibody target, and excess unbound secondary antibody is washed off. What we then do is add substrate, and substrate reacts with the enzyme and causes a colour change. So this is sort of an indication of what would happen in someone who has HIV versus someone who doesn't have HIV. Okay, in the left hand side you can see that you have the antibody from the blood attached to the antigen, then you have your secondary antibody attached. Now in someone who doesn't have HIV, you won't have the presence of the anti-HIV antibody in their blood, and as a result nothing can bind and you don't get any colour change. Okay, and the ELISA itself can be actually very basic in that the clinician is just looking for some degree of colour change. Okay, another form of ELISA is a sandwich ELISA. And this ELISA generally will involve a capture antibody, okay, that we've discussed before. So the detection antibody is present on the plate. You then add the sample, and the target of interest will bind to the capture antibody. Then you'll add a detection antibody, okay, here, which will bind to to an alternate site on the antigen, and then you can add either a secondary antibody or a substrate. So let's get a wee bit more detail on the sandwich ELISA. So generally speaking, you add the capture antibody, okay. Then you'll add a blocking agent, and the whole concept of the blocking agent is to make sure there's no binding places on the plastic plate for other proteins. But generally, the addition of the capture antibody and the blocking step will be carried out by the company. So in most cases, this is how the plate looks when it's provided to you by the company. So next step is that you have a specific target in the blood that you're interested in, and you add the blood sample, and there's a whole range of different proteins present. And the job of the capture antibody is to isolate your target of interest. Okay, and here we go, the capture antibody's done its job, and the next step is that we need to remove all the non-specific proteins that we're not interested in. So we undergo a wash and they disappear. So now we've isolated the antigen target. The next step is to add a detection antibody. Okay, so at this point the capture antibody was just to isolate the target. Now the detection antibody is going to be used for quantification purposes. So the detection antibody is added, and in this case it's already attached with a enzyme. Okay, and the next step is simply add your substrate and you get a color reaction. Another way that a sandwich ELISA can occur is that you have all the steps of capture antibody, isolate the antigen, add a detection antibody here. So capture, detection, and then you can use a secondary antibody with the enzyme attached. So the difference between the two steps here is that the detection antibody has the enzyme and it's a shorter assay. Whereas in this assay, you have a detection antibody without an enzyme, and then you have the secondary antibody with the enzyme that will subsequently facilitate the color reaction. Okay, so in some ways this is a direct sandwich ELISA and this is an indirect. Okay, and of course you add your substrate and you get your color change. Now, we talked about the blocking step, so why is it important? Because if you don't block the binding spaces on the plastic plate, as seen here, then what can happen is some of the antibodies and proteins that you add will actually stick to the plate and you'll get a false positive. Okay, so in the normal situation, when an antigen is absent, these detection antibodies should be washed away because there's no antigen. But what is actually happening without the blocking step is that the detection antibody is staying even after the wash because it's attached to the plastic, and subsequently you are going to get a color reaction. So a more common example of a sandwich ELISA is your standard pregnancy test. So we're going to start off with how the pregnancy test works when you don't have, when an individual is not pregnant. Okay, so this is the basic setup of the strip or the kit. So you have two antibodies present. Okay, this antibody is directed against the target. Okay, that is the compound that's responsible for pregnancy, whereas this antibody is directed against the detection antibody. Okay, so this is how it happens. You add your urine. Of course there's no sample, so the flow of the urine causes the detection antibody to start moving. Okay, now it's passing this antibody because there's no antigen present. Okay, so the detection antibody has nothing to bind to, and therefore the capture antibody will not isolate the antigen, the detection antibody and antigen from the solution. So they move on and they eventually get captured by this antibody. Okay, and what you then have is no binding here because there's no antigen, and you have binding here because the antibody, detection antibody, has traveled its way through and get captured here. Okay, and that's why you'll see a blue signal, one blue signal for non-pregnant when there's no pregnancy, because you're getting the enzyme reaction here but not here. Now, what happens when you are pregnant? Okay, you have the compound HCG, which is produced upon pregnancy, and you have the detection antibody. The detection antibody binds to the HCG, and the urine flow then brings these complexes along the strip. Because there is an antigen present, the capture antibody in orange is able to isolate the detection antibody and antigen complex. Okay, they bind, and then the remaining detection antibody with nothing bound to it will also bind to the bottom capture antibody. When all that happens, you then should realize that the fact that this reaction is taking place means you get blue signal and you get a blue signal. So that's how a pregnancy test works. So in this case, the detection antibody, in this case, the individual is pregnant. So next we're going to talk about a competitive ELISA. Okay. So the whole concept of a competitive ELISA is the target or the antigen that you're interested in has to compete with a labeled antigen provided by the company. Okay, in terms of competing with the detection antibody. So in this particular ELISA, the detection antibody is attached to the plate, and subsequently what happens is, again, you undergo a blocking step to avoid non-specific binding. Then you add the complex mixture of blood containing the antigen of interest. Okay, now when there's no sample present in the blood, there's no target present, then you add the blood sample, but you also add the labeled version of the target provided by the company. And when there's no sample present in your blood, the labeled target provided by the company takes up all the binding sites and you get a very high signal. Okay, so addition of labeled antigen and serum, which contains no target, means that all the labeled antigen will bind to the antibody, and when you add the substrate, you're going to get a high signal. So in summary, no or low amount of target present in the blood sample will result in a high signal. No antigen present in test sample means the labeled antigen provided by the company is free to bind without any competition or interference from unlabeled target present in the patient sample. Now, what happens when your blood sample does have target present? What happens is you add the blood sample, you also add the labeled target provided by the company, and they both have to compete for the detection antibody. Okay, so what happens is the binding occurs, then you do a wash to get rid of all excess material. And as you can see here, there are 2 labeled targets and there are 3 unlabeled targets. So quite clearly this blood sample did have some of the target that we're interested in. Okay, so the target present in the blood effectively competed with the labeled antigen. And when you add the substrate, instead of getting that really high intense signal, you get a much lower signal. And the fact that less labeled antigen was able to bind to the antibody, then there's less signal produced when the substrate is added. Okay, so the higher the antigen present in the blood means that it's able to compete with the labeled antigen, which means you're going to get a lower signal. So this is a sort of a summary of how it would look. If your sample has high antigen concentration, it's able to compete with the labeled antigen and you get a low signal. If you have low antigen concentration, then there's more chance that the labeled antigen will be able to bind to the antibody and cause a high signal. And this is just an example of how a competitive ELISA would look. The higher the absorbance, the lower the concentration of your target. Okay, and likewise, the lower your absorbance, the higher your concentration of your target. Okay folks, so this slide is to summarize the advantages and disadvantages of the different ELISAs. I'm not going to go into too much detail, but the direct is useful because it's quick and only involves one antibody step. And it also avoids any cross-reactivity of the secondary antibody because there is no second antibody in the direct ELISA. Disadvantage is the immunoreactivity of the primary antibody might be adversely affected by labeling with enzymes or tags. Okay folks, you have to appreciate that when you try to attach an enzyme to the detection antibody or second antibody, there is a risk it might affect its binding affinity. So in a direct ELISA, there's a high risk that might impact it. And a disadvantage is there's minimal signal amplification. An advantage of the indirect ELISA is the fact that the detection antibody or primary antibody isn't labeled with an enzyme, means there's a greater chance that maximum immunoreactivity is maintained. Disadvantage is you can get cross-reactivity with the secondary antibody, which can result in a non-specific signal. Okay, which again is why blocking is critically important. And the other thing is it's longer. It requires an extra incubation step because you have that secondary antibody. So advantage of a sandwich ELISA is it provides the highest sensitivity because on the one hand you have the capture antibody binding the antigen, and you also have a detection antibody binding to the antigen. It's a preferred method when the antigen is not an antibody. Okay, so it's the preferred method if you're not looking to detect antibody present in blood. The other thing is, disadvantage is requires more sample processing. In terms of competitive ELISA, generally has greater precision, accuracy and reproducibility, and disadvantage is overall lower sensitivity and specificity. So in terms of how we quantify it, okay, so we get a color change, but how do we convert a color change into a concentration? Well, basically you have to do a standard curve, okay? So for instance, you work out, if I get an absorbance of 1, what concentration will that give me? In this case, if I get a concentration of 1, I go across, I go down, and it tells me that if my blood sample has an absorbance of 1, then it has a concentration of the target of around 200. Okay, so that's how you quantify it. The color change gives off some degree of optical density, and then you're able to relate the color change with the actual concentration. And of course it's the opposite for the competitive ELISA. Sorry, it's the same, but it's inverse. So the higher the absorbance, the lower the concentration of the product. But in terms of determining when you can accept results from an ELISA, there are a few definitions you need to appreciate, okay? The first is the lower limit of detection. So here it is indicated here, okay? Here, all along here. And it's the lowest concentration at which the detection signal can be distinguished from background noise. So if you add your sample and it has an absorbance of anything here or lower, you can't rely on the value that you get, because actually it's too similar to the background. The difference now is the lower limit of quantification is the lowest standard curve point that can be accurately used for quantification. Okay, so the whole concept here is that in order to accurately determine the concentration, ideally you want it to be on this linear part of the graph. Okay, so if you get a value anywhere from here and below, you can't accurately or precisely determine or reliably determine the concentration of your blood sample or urine sample, whatever it may be. Okay, so that's a lower limit of quantification. The other terminology is the upper limit of quantification, okay? So once you get to the end of the linear part of your standard curve, after that point you can't rely on getting an accurate reading. So the upper limit of quantification is the highest point in the standard curve that can still be accurately used for quantification. Okay, so if I get an absorbance up here, say for instance, and I want to determine the concentration, I go across, I can't get an accurate value. Okay, so from using these three values, you can establish where in what situation you can accept the result as reliable. So anything, if you get an absorbance anywhere here, okay, then you can't rely on it. So this whole area of the standard curve is useless. And likewise, if you get an absorbance anywhere above here, you cannot use it to quantify the patient sample because it's above the standard curve. Okay, so essentially the region that we can work with is present in this part of the standard curve. So if you get an absorbance in your sample of here to here, you can rely on your results. So in terms of some issues you might have with the ELISA, so on the left hand side, this is the ideal scenario, okay? So you have a specific interaction between the target here and the capture and detection antibody. But what can happen is you can get cross-reactivity where some antigen that you're not interested in manages to interact with the detection and the capture antibody. And what's going to happen there is you're going to get a false positive, because when you read the results you're going to think that you are measuring this antigen when in reality you're measuring this non-specific antigen. So that's cross-reactivity. Another concept in the ELISA that you come across is interference. And interference is where your nice antigen becomes slightly modified. Okay, it might be phosphorylated or it might have some other components bound to it, which means the antibody is going to find it difficult to attach and bind to the target. And in that case you're going to get a false negative, okay? Because the antibody can no longer specifically bind to your target. Okay, another thing to consider is that the companies usually optimize this, but you need to work out what is the optimal dilution, okay? And this can have an impact considerably when you have interference factors, when you have interference. So when you do any ELISA, if you do a series of dilutions, you should always get the same result, okay? So you do a dilution and then you just use an equation to work out what the actual value is, okay? So you adjust for the dilution. So in this case it's got diluted and then got diluted, but when the individual did their calculation, the result remained the same, okay? 12, 12, 12. But the problem is when you have interference is that in a neat solution, it's the same sample by the way, but this is a situation where you have interference. In the neat solution there's too much interfering factors going on, so the companies realize, well actually we might need to do a dilution. So they do a dilution and the dilution is having a better effect, okay? So it's closer to the 12, but it's not right there yet. It's still giving a false negative of 10. But then when they did a subsequent dilution, then they established that they were able to get an accurate measurement, okay? So in this situation, the ELISA did have interference, but the company established that if we diluted to this degree, okay, fourfold dilution, we're able to get a reliable result, okay? So 12 versus 12. So that's one way that companies can overcome interference. Now in terms of controlling for errors or trying to ensure that your results are true or that your ELISA has run well, you can use positive controls and negative controls, okay? So positive controls are provided by the company, and there's a situation where you add them, and when you add them to the plate you expect to get a high rating. Okay, if you don't get a high rating, then you can basically determine that the ELISA doesn't seem to have worked as well. You also have a negative control where you have nothing. You add nothing, and if you get a response, then you can determine that the ELISA hasn't worked, because a negative control should never give a reaction. And of course you could add standards with known concentrations, okay? And that's another way to know if your ELISA's worked. And one other aspect that you need to be aware of is the coefficient of variance, okay? So this is why you always run a blood sample twice on an ELISA plate, because you want to make sure that you don't have any spurious results, and you would expect that if you add the same sample, if you measure the same sample twice, you should get the same result. If you don't get the same result, for instance, if you get over 15 to 20% difference between the two results, then you cannot accept the result, and the suggestion is you might need to repeat that sample. Okay, so what are the applications of the ELISA? Generally speaking, they're used for clinical diagnostics. So they're used clinically, but they're also used in a laboratory for research. They're, as we've talked before, they're generally, the majority of them are used to detect presence of antibody or antigen, and also used to determine serum antibody concentrations. In terms of other applications, they can be used in the food industry to detect certain toxins or components of interest. They can be used to detect food allergens present in food, and they can also be used to detect pathogens. And lastly, they can also be used for toxicological measurement of specific drugs. Okay folks, so that's an overview of the ELISA. If you have any queries or issues, please don't hesitate to contact me.